Photo 51. The most important photograph in the history of biology. The image that proved DNA's structure and unlocked the secrets of life itself. A brilliant woman scientist took it after years of meticulous work. Two men saw it without her permission, immediately understood what it meant, published their theory based on her data, and won the Nobel Prize. She died at 37, largely unknown and unrecognized. This is the story of Rosalind Franklin, the woman who discovered the structure of DNA and was written out of history. Rosalind Elsie Franklin was born in London in 1920 into a prominent Jewish family. From an early age she was brilliant, especially in science and mathematics. But this was 1920s Britain. Women weren't supposed to be scientists. Her father actually tried to discourage her from going to university. He believed higher education was wasted on women. But Rosalind was stubborn. She won a scholarship to Cambridge University, one of the few women admitted, and studied chemistry. She graduated in 1941 during World War II and immediately began research. By her mid-20s she was already an expert in X-ray crystallography, a technique that uses X-rays to reveal the atomic structure of molecules. Think of it like this. If you want to understand the shape of something too small to see with a microscope, you can fire X-rays at it and analyze the pattern they make when they bounce off. It's incredibly difficult, requires mathematical precision, and Rosalind Franklin was one of the best in the world at it. By 1951 she was recruited to King's College London to work on one of the biggest mysteries in science, the structure of DNA. Scientists knew DNA carried genetic information, the instructions for life itself. But nobody knew what it looked like. Was it a helix? A spiral? Something else entirely? Without knowing the structure you couldn't understand how it worked. Rosalind Franklin was brought in specifically because of her expertise. If anyone could photograph DNA structure, it was her. And she did. Working at King's College, Rosalind Franklin set up her equipment and began the painstaking work of photographing DNA. This wasn't like taking a normal picture. She had to purify DNA samples, position them perfectly, expose them to X-rays for hours, sometimes over 100 hours for a single image, and then develop the photographic plate. One mistake and you'd have to start over. Most of her early images were unclear, blurry patterns that didn't reveal much. But Rosalind was meticulous. She adjusted variables, refined her technique, and kept trying. Then, in May 1952, she took exposure number 51. When she developed it, she must have known immediately. Photo 51 showed a perfect X pattern, a clear, unmistakable sign of a helical structure. DNA was a double helix. This was it. The image that would unlock the structure of life. She had done it. But there was a problem. Rosalind Franklin was a perfectionist. She wanted more data, more proof, more images before publishing. She didn't want to speculate, she wanted to be certain. So she kept working, refining her findings, preparing a comprehensive paper. Meanwhile, at Cambridge University, two scientists named James Watson and Francis Crick were also trying to figure out DNA structure. But they weren't experimentalists like Franklin. They were theorists. They built models based on other people's data. And they were stuck. They tried multiple models and none of them worked. They needed better data. That's when Maurice Wilkins entered the picture. Maurice Wilkins was Rosalind Franklin's colleague at King's College. But calling him a colleague is generous. Their relationship was terrible. Wilkins had originally been leading the DNA project at King's College. When Rosalind was hired, there was confusion about whether she was his assistant or an independent researcher. Wilkins assumed the former. Rosalind knew she was the latter. The sexism was constant. Wilkins didn't respect her. He resented that she had better data than him. And he had access to her lab. In January 1953, without Rosalind Franklin's knowledge or permission, Maurice Wilkins showed photo 51 to James Watson. Watson later described the moment in his book. The instant I saw the picture, my mouth fell open and my pulse began to race. He immediately understood what Rosalind's photo meant. DNA was a double helix. He rushed back to Cambridge and told Crick. Within weeks, using Rosalind's data without her knowledge, Watson and Crick built their famous model of DNA's double helix structure. But it gets worse. Crick also had access to a detailed report Rosalind had written about her findings. Max Perutz, another scientist, had given it to him, again without Rosalind's permission. So Watson and Crick had both her photograph and her written analysis. They used her work to build their model. And then they published. In April 1953, Watson and Crick published their paper in the journal Nature, a structure for deoxyribose nucleic acid. It described the double helix and became one of the most famous scientific papers ever written. They mentioned Rosalind Franklin's data in the paper, barely. One vague sentence. We have been stimulated by the knowledge of the general nature of the unpublished results and ideas of Dr. M.H.F. Wilkins and Dr. R.E. Franklin. That's it. No acknowledgement that Photo 51 was the key evidence. No mention that they'd seen her data without permission. Rosalind Franklin published her own paper in the same issue of Nature, but it was presented as supporting evidence for Watson and Crick's model, not as the foundational discovery it actually was. So what happened to Rosalind Franklin? She left King's College in 1953. The environment was toxic and she wanted to move on. She went to Birkbeck College and began groundbreaking work on the structure of viruses. She was brilliant, productive, and well-respected in her new lab. But in 1956, at age 36, she was diagnosed with ovarian cancer. She'd been exposed to massive amounts of X-ray radiation during her years of crystallography work. Back then, nobody understood how dangerous it was. She continued working even while undergoing treatment. She published paper after paper on viruses, making discoveries that are still cited today. Rosalind Franklin died on April 16, 1958. She was 37 years old. 4 years later, in 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for discovering the structure of DNA. Rosalind Franklin was not mentioned in the Nobel Prize announcement. She was not honored. She was barely remembered. The Nobel Prize is not awarded posthumously, so even if the committee had wanted to recognize her, they couldn't. But here's the thing, they didn't want to. Even while she was alive, her contributions were minimized. Watson, in particular, spent years undermining her legacy. In his 1968 memoir, The Double Helix, he described Rosalind Franklin as aggressive, unfeminine, and difficult. He called her Rosie, a nickname she hated and never used. He portrayed her as an obstacle to their discovery, not the source of it. The book became a bestseller. And for decades, that's how history remembered Rosalind Franklin, as a footnote. A difficult woman who contributed some data. It wasn't until the 1980s and 1990s, when historians started examining the evidence, that the truth came out. Photo 51 wasn't just supporting data, it was the proof. Watson and Crick's model was built directly on her work they saw without her permission. Today, scientists acknowledge Rosalind Franklin's role. Her contributions are taught in schools. There are buildings and scholarships named after her. But she never got to see it. She never knew that history would eventually recognize her. She died thinking her life's work had been credited to someone else. So why does this story matter? Why does it still make people angry more than 60 years later? First, because Rosalind Franklin's story isn't unique. She's one of countless women scientists whose work was stolen, minimized, or credited to male colleagues. There's even a term for it, the Matilda effect, the systematic denial of credit to female scientists. Marie Curie had to fight to be recognized alongside her husband. Lise Meitner's work on nuclear fission was credited to her male colleague, who won the Nobel Prize without her. Jocelyn Bell Burnell discovered pulsars. Her male supervisor won the Nobel Prize. The list goes on. Rosalind Franklin's story is just the most famous example in modern science. Second, because Watson and Crick never fully acknowledged what they did. In later years, they admitted they'd seen her data. Watson even expressed some regret about how he portrayed her in his book. But they never said, "We couldn't have done it without her. She deserved equal credit." Instead, they've spent decades defending their Nobel Prize, insisting their theoretical brilliance was the key contribution. As if seeing someone else's data and understanding it makes you the discoverer. Third, because this is a lesson about how science actually works. We're taught that science is objective, that credit goes to whoever makes the discovery. But science is done by humans. And humans have biases. Rosalind Franklin was a woman in a male-dominated field. She was Jewish in an often anti-Semitic academic culture. She was direct and uncompromising in a world that expected women to be accommodating. And when men took her work, the scientific establishment protected them, not her. The structure of DNA, the discovery that launched modern genetics, medicine, and biotechnology, was uncovered by a woman. And history tried to erase her. If students and historians hadn't fought to correct the record, we might still believe Watson and Crick did it alone. Rosalind Franklin never got her Nobel Prize. She never got the recognition she deserved while alive. But today, every biology student learns her name. Every scientist knows Photo 51. And every time someone talks about the discovery of DNA structure, more and more people say, it wasn't just Watson and Crick, it was Rosalind Franklin. Rosalind Franklin took photo 51 in May 1952. It proved DNA structure. 2 men saw it, used it, and became famous. She died at 37, unknown. But history doesn't forget forever. Eventually, the truth comes out. And today, when we talk about one of the greatest scientific discoveries of the 20th century, we say her name. Rosalind Franklin discovered the structure of DNA. And no one can take that away from her anymore. This story made you angry? Good. Share it. Let people know. Because the best way to honor Rosalind Franklin is to make sure her story is never forgotten again. Want to learn more stories about history? Please like and subscribe.